Good day! Today, we will tackle about a plasma membrane by using a three-dimensional fluid mosaic model of plasma membrane. But before that, we will have a recap about what is cell. Cell is the basic building blocks of all living things. It composed of organelles. All cells take in food, read waste, and reproduce. The cell has three main sections such as plasma membrane, nucleus, and cytoplasm. For now, let's focus on the plasma membrane. What is the plasma membrane? The plasma membrane protects the cell from its external environment, mediates cellular transport, and transmits cellular signals. The plasma membrane, also known as the cell membrane, that separates the interior of a cell from its outside environment. The primary function of the plasma membrane is to protect the cell from its surrounding. Now, let's proceed to the fluid mosaic model. The fluid mosaic model was first proposed by S.G. Singer and Garth L. Nicholson in 1972 to explain the structure of the plasma membrane. According to them, the model has evolved somewhat over time, but it still best accounts for the structure and functions of plasma membrane as we now understand them. The fluid mosaic model describes the structures of a plasma membrane as a mosaic of components, including phospholipids, cholesterols, proteins, and carbohydrates that gives the membrane character.
The main fabric of the membrane is composed of the apophilic or dual-loving phospholipid molecules. These hydrophilic or water-loving areas of these molecules are in contact with the aqueous fluid both inside and outside of the cell. Hydrophobic or water-hating molecules tend to be nonpolar. Phospholipid molecules consist of a 3-carbon glycerol backbone with 2 fatty acids molecule attached to carbon 1 and 2, and a phosphate-containing group attached to the 3rd carbon. This arrangement gives the overall molecular area described as its heat. The phosphate-containing group, which has no polar character or negative charge, and an area called a tail, fatty acids, which has no charge. They interact with other non-polar molecules in chemical reactions, but generally do not interact with polar molecules.
Proteins make up the second major component of plasma membranes. Integral proteins, some specialized types are called integrins, are, as their name suggests, integrated completely into membrane structure, and their hydrophobic membrane-spanning regions interact with the hydrophobic region of the phospholipid bilayer. This arrangement of regions of the proteins tend to orient the proteins alongside the phospholipids, with the hydrophobic regions of the protein adjacent to the tails of the phospholipids and the hydrophilic region, or regions of the protein protruding from the membrane and in contact with the cytosol or extracellular fluid.
Carbohydrates are the third major component of plasma membrane. They are always found in the exterior surface of cells and are bound either to proteins or to lipids. These carbohydrate chains may consist of 2 to 60 monosaccharide units and can be either straight or branched. Cholesterol function as a buffer, preventing lower temperatures from inhibiting fluidity and preventing higher temperatures from increasing fluidity.